After studying this module, you shall be able to know the various properties that enzymes possess, like differences in their size, charge and solubility. You are going to learn about how they are extracted from cells. You are going to know about various tools which are utilized to extract and purify these enzymes. Let's learn about the purification of enzymes. Remember, any functional study of enzymes cannot be performed until we obtain their homogeneous preparation. When I say the word homogeneous preparation, it means that it should be a pure enzyme without any contamination. Now, this homogeneity of enzyme is very essential in all its applications, like therapeutic use. Enzymes, being proteins, are separated on the basis of differences in solubility, charge, size and various other ways. Cell is broken to prepare what is known as the cellular extract, or commonly known as crude extract, bringing the enzymes in solution, which is then separated from the other proteins, let's call them contaminants here, on the basis of physical properties like size, charge and solubility. Now let's look at what is the protein purification strategy. Most enzymes, as we know after reading the previous modules, that most of the enzymes are proteins. Now the purification and extraction methods employed for enzymes are same as that of proteins, because enzymes are essentially proteins only. To study an enzyme's function, it is essential to obtain its what is known as a pure preparation, a homogeneous preparation from cells which contains multitude of other proteins and even enzymes. Plethora of enzymes which are present in the cell varies with regards to their molecular weight, charge, size, etc. Now these differences form the basis of separation of one enzyme from the others. First consideration which is to be made is the source of the enzyme. For example, suppose we want to extract out acid phosphatase. So the best source would be germinating seeds, which are a rich source of acid phosphatase. They are preferably used as a source for extracting acid phosphatase than the ungerminated seeds. Why we are saying ungerminated seeds? Because the germinating seeds are going to have more proportion of the desired enzyme. Thus, the source chosen should be the richest source of an enzyme. Once the source is decided, the next step is to break open the cells so that the proteins now come into solution. This is called the crude extract. If the enzyme is a mitochondrial enzyme, let's take an example, which is localized or it is localized to any other subcellular organelle, differential centrifugation is generally performed to enrich the enzyme in the crude extract. Crude extract is then subjected to what is known as fractionation. Fractionation is basically a process that separates the proteins from different fractions, again based on their size, charge or solubility. We are going to first study what is the first and foremost fractionation procedure which is commonly followed. This is generally known as a salting out phenomena. It basically, what it does is it utilizes the property of differences in solubility. Remember, the physical property being exploited here is the solubility. Now, addition of specific amount of salt precipitates some proteins, enriching the enzyme of interest in the preparation. The salt which is generally used in this procedure has to be possessing the high solubility in water. So it is generally ammonium sulfate or potassium phosphate. Any protein in solution, we know that interacts with the solvent molecules by virtue of polar interactions. Now, addition of a small amount of a salt increases the solubility of a protein, because as the salt concentration in the protein solution increases, additional counter ions are now present to basically shield these ionic charges of the protein and hence increase the solubility in solution. So basically this is called a salting in procedure. Salting in means you are giving minuscule amounts of the salt, which in turn is increasing the solubility of protein. However, if you are going to further increase the amount of the salt, if it is a high amount of salt, is going to cause predomination of the solute-solute interactions than the solute-solvent interactions. So, solute-solvent interactions basically predominate, causing the protein to now precipitate. The increase in salt solution is thus making solvent limited for the dissolution of the solute. Thus, this is a dehydration reaction. That's why it is very important that the salt is to be added really gradually so that dehydration proceeds slowly. If, for example, if you are performing the salt addition very fastly, the dehydration is going to be really faster, which can even denature the protein. So, the addition of salt has to be a gradual addition of salt. The precipitated protein, remember, is now going to contain excessive amount of salt. So, this precipitated protein is then resuspended in an appropriate buffer containing a suitable pH. Now, remember, this buffer and suitable pH are the parameters which are being decided what is the kind of protein which is present. It should be, its structure, its function should be retained in this particular buffer and pH. The resuspended solution is then dialyzed to remove high salt concentrations. This particular figure, as you can see, depicts the procedure of dialysis. The resuspended solution has been taken in this dialysis bag. Dialysis tubing is basically a semi-permeable membrane. Now, why we are calling it a semi-permeable membrane? Because it is going to allow certain things to move in and out, but not the other things. Now, what happens is whenever you have resuspended, taken this precipitated protein into this dialysis tube, this is then suspended in a larger volume or of less concentration of the buffer. The dialysis bag then allows only the small molecules such as the solvent to pass through, but not the proteins, which are like much bigger in size and hence retained in the bag. As you can see in this figure, the solute is moving out from a high to a low concentration till the concentration reaches a equilibrium. Other agents that can be used for protein precipitation are inorganic salts, organic solvents, change in a pH, or polyethylene glycol. Now, this dialyzed protein solution is then subjected to further fractionation procedures. The next method we are going to study is the column chromatography. Now, what basically column chromatography, if it is going to utilize the property of size, it will be called a gel chromatography. The other physical property which is utilized is the charge. In that case, it is called the ion exchange chromatography. Affinity chromatography, on the other hand, can be used if any inhibitor for the enzyme of interest is known. All these column chromatographic procedures use a buffered solution, the mobile phase, and the porous solid material held in a column which is called the stationary phase. Basis of these chromatographic procedures we are going to study in this particular module. Let's learn about the gel chromatography. As I just mentioned that gel chromatography is basically going to utilize the physical property which is the size of the molecule. So, gel chromatography is also in turn known as size exclusion chromatography or molecular sieve chromatography. We shall learn why these two names are given to gel chromatography. It uses the property of size of protein to separate it from the other proteins. Now, the solid phase in this chromatography basically consists of beads of with pores of defined size. Suppose it is given a name G50 gel matrix. So, this G50 gel matrix is going to possess a pore size cutoff of 50 kDa, which simply means it is going to allow the proteins of less than 50 kDa to enter the pores, and hence these proteins will emerge later from the column, while the proteins with greater than 50 kDa molecular weight do not enter the beads and hence are going to follow the shorter route and emerge earlier from the column. We can see in this particular figure what I just mentioned, larger proteins emerge out earlier than the smaller proteins, because the smaller proteins are going to enter the gel matrix and they are going to traverse the more path compared to the larger ones. So, the larger ones, they are moving faster compared to the smaller ones. Now, this was with regards to size exclusion chromatography. Now, let's learn about ion exchange chromatography. Now, this is our diagram which is depicting the ion exchange chromatography. Ion exchange chromatography can be of two types, an ion exchange or cation exchange, depending upon whether it is the positive charge being exchanged or the negative charge being exchanged. So, this ion exchange chromatography basically separates the two proteins on the basis of charge. As I just mentioned, there are two kinds of exchangers. It can be cation exchangers or anion exchangers. Now, a cation exchanger itself is negatively charged. Remember, we generally confuse this. Cation exchanger itself has to be negatively charged. When the protein solution is added on top of the column, the movement of positively charged proteins is impeded because of its interaction with these negatively charged exchanger, while the negatively charged proteins move faster. Thus, two proteins with opposite charges make two distinct bands and eluted in different fractions. An ion exchanger, if we see in this particular figure, itself is positively charged, but what it is going to exchange is the negatively charged molecule, which is the anion. Positively charged analyte is however exchanged by the cation exchanger. So, this is in terms of what we wanted to learn about ion exchange chromatography. Ion exchange chromatography, remember, is going to utilize the charge, while the one we learnt earlier, whether it is molecular sieve or we call it gel chromatography, it utilizes the size of the molecule to be separated from the other contaminant or from the dialyzed protein. The next procedure which is generally followed is the affinity chromatography. Now, affinity, as the word suggests, there has to be affinity between we know certain property of the molecule which we want to separate, or the protein or enzyme we want to separate. We know to which molecule it has affinity with. So, affinity chromatography basically relies on the basis of affinity of a protein with its ligand. For enzymes, best ligands which we know of to be used for affinity chromatography are basically either the poor substrates or inhibitors. Now, why they can be poor substrates or inhibitors? Just because poor substrates are going to interact with the enzyme longer compared with the good substrates. In this column, you are going to see that the stationary phase, the ligand is attached to this particular stationary phase, which possesses, this ligand now has the affinity with the enzyme which we want to separate. When a mixture of proteins is poured onto the top of the column, it is going to gradually enter the stationary phase, basically interacting with the ligand which is attached to it. So, what we are going to see is the enzyme of interest with affinity for the ligand binds to it, while others do not have this affinity for the ligand. So, they are going to just come in the flow through. Then the higher concentration of free ligand is added onto the column, and as we know that because of the competition, enzyme is going to now bind to the free ligand and is going to get eluted out. In this figure, we can see the outline of how basic steps of affinity chromatography are performed. The mixture of proteins is loaded onto the column. The protein mixture is which is added, the stationary phase contains our ligand for our enzyme which we want to separate. So, this only a selected protein is going to bind to the ligand and rest are going to come through in the flow through. and after this, when we want to elute out and retrieve our protein from the column, a very high concentration of the free ligand is added and we have the elution of the our specific enzyme. So basically in this particular module what you have studied is most enzymes are proteins and hence same procedures are employed to extract proteins as well as enzymes. The extraction procedures use differences in charge, solubility and size for separating the protein or enzyme of interest from other proteins. Now the first and foremost procedure which is used in this particular, the purification, relies on the basis of differences in solubility of enzyme in a salt solution. Ammonium sulfate is preferentially used because of high solubility in water. Next, the dialysis is performed to get rid of the excess salt. Following dialysis, one of the column chromatographic procedures are performed, namely it can be ion exchange chromatography relying on the physical property which is the size. In case of ion exchange, it is the charge which is exploited. Next is the size exclusion chromatography, in which we are going to utilize the size of the physical property, size of the particular protein or enzyme. And last is the affinity chromatography which we just studied. Affinity chromatography relies on the affinity between the molecule to be separated and the ligand. This is to separate and purify protein and enzyme of interest from other contaminants or other cellular proteins.